Lab 2, we are also going to do diffusion, osmosis, okay, and look at the movement of materials into and out of the cell itself. Now we are going to be using, for this osmosis lab, when osmosis is the diffusion of water across a selectively permeable membrane, which is a biological term because the selectively permeable membrane is the cell membrane. We are going to be using dialysis tubing for that. And you can look at this. This is a piece of dialysis tube that is deflated at this point, has nothing in it. We've tied off one end. We're going to fill it with a solution, and then we're going to clamp the end with this little barrette clamp, and we're going to leave it in another solution surrounding it. We're going to set this up with 4 different beakers. The 4 different beakers are going to have different solutions. The first beaker, which has already been set up, has the dialysis tubing filled. It is filled with with glucose. Glucose is a monosaccharide, and we have placed that glucose-filled sac into a beaker full of distilled water. So we are placing it into a hypotonic solution. Once we had filled it, then we weighed it initially, and at the end of the lab, we will also weigh it again to see if it has gained or lost any weight. Now remember, osmosis is the movement of water into and out of the cell membrane. So this being example of the cell, the water will either move in or out. And in the hypotonic solution, which means there is more water, less solutes outside the cell than inside, the water should move in. So we should see an increase in weight in that sac. The second sac we have filled with glucose, 40% glucose, and we are going to surround that with 40% glucose. So we're going to put that in the beaker, and we're going to leave it to run for about an hour and a half, and we are going to evaluate whether the sugar moved in or out, whether the sac has gained weight. We've weighed this sac prior to the beginning of the lab. We'll weigh it again. Now this is representative of an isotonic solution because it has the 40% glucose inside the cell as well as outside the cell. So ultimately we should see no real net change in weight because there is movement into and out of at the same exact rate since it is isotonic. The third beaker we have filled with distilled water, and in the sac we have filled it with a sodium chloride solution. This is a salt solution, 10% sodium chloride. This is also going to be a hypotonic solution relative to the sac. And the last one is the beaker. We have it surrounded by distilled water, and we have in this solution sucrose, which is a disaccharide. It is bigger than the glucose, which is a monosaccharide, and this is mixed with a dye called Congo red. So we're going to look to see if the sugar can leave the cell and if the dye, Congo red, can leave the cell. So we're going to fill this sac up with 20 mLs of the fluid that we have already measured out. We're going to clamp it. We would then get a weight on the scale, and then we would submerge it into the fluid and we will leave it alone. Now this will run for about about an hour, hour and a half. And then at the end of that hour and a half, we would then retrieve our beakers, okay, and we have done so here. We're going to show you the end results. And we're going to look at the different beakers and the different solutions. Now, we have in beaker 1, after it's run for an hour and a half, it started out, it was flaccid and limp, okay? But at the end, it has gained much more water, and because it is a hypotonic solution and the water has been able to move into the sac, it has gained weight. So we would weigh this, we would see what the difference is between the final weight and the initial weight. That would give us how many milliliters it gained during that time period, or how many grams it gained during that time period. And then we would divide it by the time period to get the actual movement and diffusion rate of milliliters per minute, how much it gained. Okay. Now we are going to look at number 2. Number 2, remember, is in the isotonic solution. Sucrose in the sac, sucrose in the beaker, and we should see no real change. Okay, maybe a little bit of change, not much. Again, we'd weigh it, find out that there was very minimal weight gain, so that means that the solution did not move into or out of the sac at any particular rate. Number 3 is surrounded by distilled water and it's sodium chloride inside, so it is in a hypotonic solution, and this will also have gained some weight because, again, the water is moving into the cell to dilute the fluid in the cell. And the last test tube, the sucrose in the Congo red, is in an isotonic solution. It too will have gained weight and plumped up, and there will be a net weight change. Okay. So the ones that are in the hypotonic solution, that would be 1, 3, and 4, all gained weight, and they look much bigger. So we have a weight gain. We can divide that change in the weight divided by the time that it was run, and we can get a rate of milliliters per minute, or in this case, milliliters per hour of weight gain. Okay. Now, the other thing we're going to do is that we want to not only be able to say, okay, the water moved, but what about the sugar? In number 1, we had sugar, the glucose, in the sac, but not in the beaker. So we're going to test both the sac and the beaker solutions at the end of the lab, and we're going to test them with Benedict's. Benedict's is an indicator of the presence of sugar. It will start out blue, and you have to boil the solution, and then it will turn any color besides blue to indicate there is sugar present. If it stays blue, that means there's no sugar present at all. So when we did do that experiment, okay, we are going to see that in the beaker and in the sac, they both indicate that there is sugar present. Now, we started out with no sugar in the beaker fluid, but there's sugar now at the end of the lab, which means that the sugar was able to diffuse out of the sac into the beaker. Now, diffusion is going to be the movement from high to low concentration with no energy involved, and it is going to be a non-biological movement. So when we talk about the sugar moving from high to low, we call it diffusion. But when we talk about the water moving from high to low across that selectively permeable membrane, the sac, or the cell membrane, we call that osmosis, or we can say that the water osmosed into the sac. Now, for the number 2 setup, it was the same thing in the sac and outside the sac. Both had sugar, the glucose, 40% glucose. So there's no need to test because there should be glucose, should be positive for both of these. So we're not going to even test those. For test number 3, we are going to, I wonder where my silver nitrate is. We're going to test for the presence of the sodium chloride to see if the sodium chloride moved out. And with that, we will test with some of the solution in the beaker because the sodium chloride started in the sac. And we're going to put a drop of silver nitrate into that test tube that we have with the fluid in it. And we'll see it will change into a white precipitate. Okay. And then for test 4, we have sucrose that has in our sac. We're going to test for the presence of sucrose in the sac. The negative part of this is, though, remember, sucrose is a disaccharide. It's considerably larger. So when we test for the presence of sugar in the solution, we end up with a negative test. The negative test, that is still blue, of Benedict's indicates that no sugar was able to move out. And the reason for that is that your selectively permeable membrane is like a screen. It has holes in it, and small particles can move through, but if you get a big particle, it can't move through. Glucose is small, it's a monosaccharide. Sucrose is a disaccharide, it's too big to move out. In addition, we had no movement of the red solution, so the dye is also too large to move out. Okay. Unfortunately, there's no color. Okay, I'm grabbing the silver nitrate so we can actually perform the silver nitrate test on the fluid from beaker 3. Okay. And you'll see as you drop 1 or 2 test tubes that there is a white precipitate being formed here, which indicates the positive test for the presence of the sodium chloride. Actually, it's silver chloride is forming. And that means that the sodium chloride was able to also pass through that selectively permeable membrane. It is much smaller, breaks down to little ions, and it can pass through. So the net result of this experiment is that, one, when we put them in a hypotonic solution, there will be osmosis of the water into the sac. And the second thing is that the Benedict solution, to indicate whether or not the sugar moved out, was positive for the beaker solution when we had glucose in the sac, but it was not positive when it was sucrose in the sac, because sucrose is too large to move.